Okay, well, welcome everybody to day 3 of the Euglena International Conference. We've had a great couple days so far. Today should be no different. Some great speakers on the docket. I'd like to introduce Dr. Anna Karnkowska from the Institute of Evolutionary Biology in Warsaw. Anna is a professor in the Institute. Her research focuses on the diversity of evolution and ecology of eukaryotic microorganisms, one of which is our friend Euglena. Without further ado, I'd just like to hand over. Anna will be chairing the session this morning. So over to you, Anna.
Thank you. And it's a great pleasure to chair this session, which will be focused on evolution. Obviously, with our main focus on euglenoids and our understanding on evolution of this group, as well as how we can use this group to understand bigger evolutionary questions. So yesterday and the day before, we learned a lot about, well, first of all, the diversity of this group, the ecology, and how resistant they can be for various, well, environmental and in different environments for different features of these environments, as well as to some extent on what we know already about their genomes, in particular, obviously, Euglena gracilis, and the challenges which are ahead of us in the experimental work. And of course, to answer these ecological and evolutionary questions, we have to move forward the field and work on the euglenoids to use other techniques. So I think there were some nice talks on how we can use already these techniques in different groups of algae. Hopefully, we will be able to apply them also for euglenoids. And as already mentioned today, we focus on the evolution. We'll have 2 sessions. And in the first session, we have three speakers, and starting with Vladimir Hampel, from the Department of Parasitology at Charles University in Prague. Also, I have this pleasure to mention that Vlada was my supervisor during postdoc. So, but I'm super curious what happened since I left, and some things I know, but I'm very interested what's new in euglenoids happen in his group. And Vlada will be talking today about evolution of plastids in this group.
Thank you very much, Anya, for kind introduction. Hello, everyone. I am very proud that and honored that I can start today's session. And I hope you can see my screen now.
Yes.
Looks good.
Okay. So, as Anya already said, I will talk about the things related to euglenoid plastids, but actually it will not be about the function of the plastids themselves, but rather about the consequences the origin of plastid and euglenoids brought to the euglenoid genomes and euglenoid functions, let's say. And I would like to start actually with the acknowledgement, because the data that I will present in the most part of my talk were generated, of course, and analyzed by other people than me. So this is a rather older picture from
I'm sorry, but
my team.
I think we don't see you switching the slides. At least I don't.
So maybe I'll stop share and share again. So I will share the whole screen.
Yes.
So how is this?
Yes, yeah. and can try to switch now the slide.
And now?
Yes, it's good.
Okay.
Thank you.
Okay, thank you for telling me. So this is my team in the past, and several people were analyzing and generating the data that I will present today. Stepanka, Peter, Anna, Kika Zahonova is not present in this picture, but she also took part. And I would also like to thank to several people who kindly generated the data sets that we analyzed. That's Ania Karkowska, our today's chair, now Jiu Buki, Rafał Milanowski, Magdalena Plecha, Gordon Lax, and Alastair Simpson. So with this, I would like to start, and as I already said, I will focus on chloroplasts in Euglena, and it's been very early noticed by Sarah Gibson already in 70s that chloroplasts in Euglena are special, like different from chloroplasts of green algae. And the morphological specialty is very nicely shown here on these electron micrographs. And that's the envelope, which composes of 3 membranes as opposed to 2 membranes that are known to green algae. And it's been Sarah Gibbs who right away in this paper in 78 proposed that the reason might be that these chloroplasts didn't originate from the primary endosymbiosis of cyanobacterium, but rather from a secondary engulfment of an alga, already of eukaryotic alga, likely a green alga. And this has been then later proved after the chloroplast genome of Euglena has been sequenced and later analyzed by several people who focused on reconciliation of the phylogenetic origin of the chloroplast. And as I said, it's been, I noticed here 2 main papers that I would like to cite in this respect, and the phylogenetic origin of chlorophyte, of euglenophyte plastid is nicely shown in this 3 figure where Jackson et al. In 2018 placed it inside the green algae sister to the lineage called Pyramimonadales. So, and that's basically the home from which the Euglenoid plastid has been derived. So the fact that the organelle has been acquired by endosymbiosis from green alga related to nowadays prasinophyte alga Pyramimonas, as drawn in this picture, has an important consequence. During every endosymbiotic event, and we know it now very clearly, some genes are transferred from the endosymbiont to the genome of the host. And in the secondary endosymbiotic event shown here, where the red eukaryotic secondary host engulfs the primary host, Pyramimonas alga, the gene transfer happens not only from the plastid itself, which is also possible, but primarily or mainly from the genome of from the eukaryotic genome of the green alga. And obviously people investigated this phenomenon also in Euglenoids along with other secondary and primary algae. And several people in the past, whose papers I note here, noticed that the Euglenoid datasets, they focus mainly on Euglena gracilis, but in some cases also on some other Euglenoids, contain genes that are affiliated to algae. And this is probably best exemplified by the latest study of Ponce Toledo 2018, from which I have taken this figure, which did it, I would say, most systematic in a most systematic way, and they recovered slightly over 60 genes from Euglena gracilis genome, which in phylogenetic trees were clearly affiliated above a very strict bootstrap threshold with either green, most of them with green, or with red algal groups. By the green algal groups, I mean, or they meant green algae itself, but also chlorarachniophytes, which were derived from, whose plastid was derived from green algae. By red algal groups, they mean either primary rhodophytes or groups that have red lineage plastid. So from this picture, it is so this illustrates one interesting phenomenon, which has, however, been reported also from some other cases of algae, of secondary algae. And this phenomenon, and by this phenomenon, I mean that although we know that the current plastid of euglenophytes is derived from green algae, we see in their genomes quite large proportion of red algal genes. And this has been shown also for chlorarachniophytes. And in the opposite fashion, for secondary red algae, where also some quite large proportion of green algal genes has been reported, which is again not expected from the fact that they have undergone endosymbiosis with red algae. So these observations have been explained by a hypothesis proposed by Chris Hove. This model is called chopping bag model, and basically it reflects to it proposes that although we now observe red or green algal plastid in these lineages, they have probably undergone in the past cryptic endosymbiosis or temporary associations with some other types of algae from which they acquired their genes, which are now stay in their genomes. So we have decided to reanalyze the data for euglenids, and particularly, I would like to explain what I mean by that. Pay great attention to the evolutionary context of observation of certain number of green and certain number of red genes in the genome, because we believe that the context is actually very important for the interpretation of the data. We took part, so this is a one drawing of phylogenetic tree of euglenids, which illustrates the fact that has not been that much stressed at our conference so far, that besides the green euglenids represented by Euglena gracilis, most of the talks been about. So besides the green euglenids, the euglenid group contains also a whole lot of heterotrophic euglenid species that are depicted here, from which the Euglenophyta, the green euglenids, originated, presumably in a single event by acquisition of the green algal plastid, as shown here. And so in our work, we took part on 2 genomic projects of euglenids, one of Euglena gracilis, that's a Benazer et al. Paper, and our paper of Rhabdomonas costata genome, which is a heterotrophic euglenid. And of course, we were doing several things during the genome annotations, but one feature that we were focusing also was the origin of genes in these genomes. For that purpose, we have generated a phylogenetic pipeline through which we run all the genes found in these genomes or transcriptomes. I will not describe it, well, I will describe it only very briefly. It's nothing super new. Basically, each gene in this organism, one by one, was blasted against a local database composed for this purpose, aligned with its homologs. The alignments were trimmed, trees were constructed and bootstrapped, and then the topologies of the trees were automatically sorted based and binned into various categories based on the taxonomy of the closest relative of the Euglena sequence, Euglena or Rhabdomonas sequence. So the binning, I would like to explain in a slightly bigger detail. So basically, we bind those trees in which the sequence in question, so that's the one sequence of either Euglena or Rhabdomonas, depending on the project, was sisterly related to a taxon with a homogeneous clade or a single taxon with bootstrap support of 75% or higher. So I can illustrate it, for example, on this example tree, in which Euglena gracilis sequence branch actually nested within chlorarachneophyte sequences. So all these in the green rectangles are chlorarachneophyte. And the bootstrap support for this clade was in our analysis 74, which means that actually this tree was not binned into any bin because it didn't reach the threshold 75 and was discarded. But if the bootstrap would be higher, 75 and higher, it would be binned into a group called chlorarachneophyta, meaning that this particular Euglena gracilis gene was affiliated to chlorarachneophyte sequences. So this is roughly our methodology. And here are the results which were generated individually for Euglena gracilis and Rhabdomonas costata and published in the respective genome papers. And so what you can see on the y-axis is the number of proteins that were binned into respective categories. Categories in the shades of gray or black are this excavata, discova, kinetoplastid are basically reflecting the vertical origin of genes, so sister groups of euglenophytes or where euglenids belong. While the colorful columns represent bins of groups which are unrelated to euglenids, and so affiliation of euglenid genes to these groups is remarkable in some respect, and probably reflects either some horizontal gene transfer contamination in the data set or some phylogenetic artifact or something. So what you can see if you compare these two graphs that were generated by exactly the same methodology from two organisms, is that in Euglena gracilis, these first colorful columns are a little bit higher, which denote viridiplantae haptophytes and like secondary red algal groups, which you don't see here in Rhabdomonas costata. But you can also see here, and this is where I'm coming to the context that I mentioned at the beginning, that we have binned the genes not only to algal groups, but also to several non-algal groups like metazoa, choanoflagellates, fungi, amoebozoa. And these not algal bins, for which you have no reason to expect genes to be present in these euglenic genomes, they can define for us some threshold, let's say some level of noise that our method has, because every method, of course, is prone to errors, and you have to be aware of that. So, for example, the height of the columns in metazoa and in amoebozoa could tell us that if the proportion of those genes is below this number, you are in a gray zone where this where the presence of such genes can mean something, but you have to interpret it very carefully, because it can also be just the artifact or noise of that method. So suddenly we see that only the algal slopes, viridiplantae and secondary red algae, go above this threshold, and in Rhabdomonas costata maybe we see picking out the slope of the column of viridiplantae. And here comes the other type of context that we would like to investigate, which can help us to determine whether this slightly enriched Viridiplantae column in Rhabdomynas costata is anyhow meaningful, or random fluctuation in this particular species. And this broader context means including more Euglenid species. So we went further and sampled with the help of our collaborators and frank donors, so of datasets, sampled the datasets of other euglenids. We included another Euglena species, Euglena hyemalis, secondary heterotrophic Euglena longa, Eutroxella gymnastica, mixotrophic Rapaza viridis, and 3 more heterotrophic euglenids. Most importantly, we have included also 10 non-euglenid species as outgroups, which will also provide the context that we need for interpretation of our data. With these data sets, we rerun our pipeline, which I explained already. So nothing has been changed. And here I would like to know that we have actually here performed 2 types of binning, either the binning that I have already described, and more, let's say, strict binning in which to bin the sequence, it was required that the sequence was actually nested within the taxonomically uniform clade with bootstraps, with 2 nodes with bootstraps over 50%. Results of these 2 binnings are very similar, and I will now show data only from this sister-related relationship binning shown on the left side. So this is a graph summarizing presence or proportion of genes in various genomes of euglenophytes and outgroup taxa. On the x-axis is a proportion of what these genes take from the data sets. So we have always calculated, we have recorded not the number of genes, but the percentage of those genes from the total transcriptome or genome. And here in this legend, you can see the color code for the groups. So obviously green are Viridiplantae, red are rhodophytes, shades of brown are secondary red algae. After a while, or very soon, very soon, the opposite. Very soon we have noticed that the dataset of heterotrophic Euglenid Pulatia is likely highly contaminated, and so we excluded it from the analysis. Now we decided to take advantage of the fact that we have in the analysis several species belonging into the same, let's say, ecological group of euglenophytes, that we have there 3 autotrophic euglenophytes, 3 heterotrophic euglenophytes, and outgroups from the group of Discoba and Amorphea. And we set up to test whether the differences in the content of various types of genes are significantly different between these groups. So obviously this, I will not present the data of all the types of genes, but focus only on green and red. So first, I am presenting a graph summarizing the results of the green algal content in those various euglenophytes. Here on the right is the situation in Rapaza viridis and Euglena longa that couldn't go into the test because they are just a single in their group. They have we treat them as a somewhat special because Rapaza is mixotroph and Euglena longa is secondary osmotroph. So we didn't include it into the test groups. The red column are genes related to Viridiplantae, the light, green column Viridiplantae, light green chlorarachnoids, and the black is the genes from the green lineage in whatever green lineage, which means either Viridiplantae, chlorarachnoids, or a mixture of both, if the phylogeny was complicated. So what I would like to stress here, that it is obviously seen that the amount of green algal genes in eutotrophic euglenids it's statistically significantly higher than in heterotrophs, which is shown here, and statistically significantly higher than in the outgroup taxonomic groups. The same applies also with lower significance for chlorarachnoids. However, the heterotrophic euglenid gene, green gene content basically didn't differ from the outgroup taxa, which rejects any significance of that small increase of green genes that we have observed, for example, in the Rabdomonas costata graph I presented a few slides before. So there is no significant enrichment of green genes in heterotrophic euglenids. Now to the red part. So here, red the graph is the same, has the same setting. The red column are rhodophyte genes, shades of brown, genes of various secondary red algae, and the black column means that the gene is coming from the red algal lineage. So one of these four or a mixture of these four. Again, Rapaza viridis and Euglena longa are on the side. And you can see the situation here is a bit different, that there is significant enrichment of these genes in autotrophic euglenids compared to the outgroup taxa, but not compared to the heterotrophic euglenids. And moreover, the heterotrophic euglenids seem to have less significantly, but still significantly on a lower statistical support level, enriched red algal content compared to the outgroups. I forgot to mention on the previous slide that situation in Euglena longa was always the proportion of these genes, both red algal and green algal in Euglena longa was always a bit smaller than in eutotrophic euglenids, and in Rapaza viridis on the opposite always higher than in eutotrophic euglenids. But we couldn't include this into the tests because they are not part of the test groups. So then we performed another type of analysis, which I will not go too much in detail by methodology. But unlike the previous analysis, where we analyzed always a single euglenid gene at a time, here we grouped the genes in orthologous groups and analyzed the orthologous groups, did the phylogenies of the whole orthologous group. And we're selecting such a topologies where more than 2 euglenid sequences branched with some unrelated taxon with bootstrap support higher than 75, and bind them in a similar way as previously. The presence of more than 2 euglenids allowed us to map acquisition of these genes, or putative acquisition of these genes. Of course, other scenarios are also possible. Putative acquisition of these genes onto the euglenid tree using Dolo parsimony, and I will show you the result on this slide, which is the last slide of my presentation, actually. This is kind of a busy slide, so I'll walk you through it. So what you can see on those, so in blue is like a skeleton phylogeny of euglenid taxa that we were analyzing. And on these inner nodes in the column, you can see the number of genes, the legend, or like the bar to measure how many genes were placed into those nodes is shown here. So here the head of the column and means the number of genes from that particular bin. So this is a bin of green lineage genes, which was placed by Dolo parsimony in this particular node of the phylogenetic tree. The columns going up were sorted into larger 6 bins: green lineage, red lineage, glaucophyte, and a mixture of any algae. That's the gray. While the columns going down were sorted more like more finely into the viridiplantae, chlorarachnioids, rhodophytes, ochrophytes, optophytes, cryptophytes, oof, I forgot these two. So most events were actually placed on the species-specific branches, and these are not shown here, but those which were placed on the inner nodes are shown here. So what you can see here, that the trend is definitely not clear-cut, but there is one node which is the common ancestor of Euglenoids, so common ancestor of Euglena and Eutreptiella in this particular case, where you can see that there is increased where the influx of green algal genes was raised, while before the influx of the green algal gene was much smaller. While the influx of the red algal genes doesn't change, let's say, that much, at least from the first look. So to sum up these analyses that I have, that we, not me actually, that we have performed and that I try to present, I would like to say that we have determined the proportions of genes with algal affiliations in multiple euglenoid species with various trophic modes. And in the context of, and we put it into the context of non-euglenoid taxa. We have also applied statistics to compare the number of the proportions of genes between euglenoid groups to see which numbers are actually significant and which are not, or which are increases are significant and which are not. And with this, we have confirmed that the Euglenoids, the green euglenoids, indeed contain significantly higher proportion of green algal-related genes, which likely reflects the endosymbiotic event which happened in their common ancestor. While the proportion of genes from the red lineage, they are present there as they have been reported in the past, but their proportion in green lineage, in green Euglenoids, does not differ significantly from heterotrophic euglenoids. So there is not significantly increased, although there is increase, it is not significant. And quite surprising, or I don't know, surprisingly, it seems generally higher in Euglenidae, so in both auto and heterotrophic euglenoids, as compared to the outgroups, at least to the outgroup that we used in our study. So the reason for this is unclear. Maybe there was really some cryptic red endosymbiosis, or at least temporary association of euglenoids with red algae, or they simply like red algae or red algal groups, red algal plastic containing groups as their food. Generally, the proportion of algal-related genes in Euglena longa, the secondary osmotroph, is decreased, which must be taken into account in interpretation of the data in in heterotrophic euglenoids, because definitely with the loss of photosynthesis, the proportion of algal-related genes goes always down, or seems to go down. I don't know if always, but in Euglena longa. So it would be interesting to analyze other cases. While mixotrophic Rapaza viridis contains extremely high proportion of algal-related genes, the highest of all euglenoids that we have investigated. So with this, I would like to end my talk and thank all of you for listening to me, and again thank people that analyzed or provided the data.
Thank you very much, Vlada. And of course, the questions will be, everybody can post the question on the chat and we'll discuss it after all the talks in the session. And now we'll be switching to the next speaker, second speaker in this session, Pavel Halakuc, who is a PhD student in the same institute as I'm working, so Institute of Evolutionary Biology at the University of Warsaw in Warsaw. And Paweł will be also talking about organelles, but this time we switched to mitochondria.
Thank you for your kind introduction. I hope you can see and hear me well.
Yes.
So good afternoon, everyone, although it's pretty dark already in Warsaw. I'm Paweł. I'm a PhD student at University of Warsaw, and I'm very excited to be able to share results coming from my very first own grant. So as the short introduction, let me tell you about mitochondria. So you probably all know that they are semi-autonomous organelles coming from endosymbiosis with alpha proteobacterium. And one of the remnants of their bacterial past is that they contain their own genome. And we usually picture it somewhat like that, based on our bachelor studies. Mitochondrial genome, human mitochondrial genome is a relatively small circular molecule, which encodes about 2,000 of genes. But when we look across the whole diversity of eukaryotes, we can find that mitochondrial genomes are almost as diverse as the eukaryotes themselves, both in structure, when they can be linear, circular, fragmented, or compact, and in gene content, which also varies a lot across eukaryotes. Our favorite Euglena gracilis is right here, and you can see that it's rather on the lower end of the gene number spectrum. And in fact, its sister clades, like Indoplastin Plenemids, are known for rather large genomes, which encode relatively few genes. And in both those groups, complex systems for maintenance of the genome and gene expression have been described. However, nothing like that was found in Euglenids. In fact, only Euglena gracilis have been well studied, and several times presence of short linear chromosomes have been reported. And it is consistent with our own study, in which we used both short and long reads from 3 closely related Euglena species to determine how those mitochondrial genomes look like. And in all 3 cases, we have identified genes present in multiple copies. And within those copies, only the gene and its closest neighborhood is well conserved, while the long molecule tails, they can be over 50 kilobase pairs, are very divergent. And those tails contain a lot of short gene fragments of various mitochondrial genes, and they are usually 50 to 200 nucleotides long. So the question I asked myself, looking at those, was whether this picture is typical for all other euglenids. The diversity of euglenids you can have seen, for example, on Vladia's presentation. And it's difficult to imagine it looks like that in all of them. And so I decided to check whether this multiple linear chromosome structure is typical for them, whether the genes they encode are the same. And based on those 2 answers, how did the mitochondrial genome of ancestor of all euglenids look like? To answer those questions, I've used genomic data coming from phototrophic euglenids. For now, I'm working with green euglenids because they are easier to cultivate and to work with than heterotrophs. I sequenced total DNA coming from single cells oil cultures, assembled the genomes, and used various methods to identify mitochondrial contings within those. And the most crucial part of my work was employment of the small software called Bandage. Bandage allows us to visualize the brine graphs. For those of you who are not bioinformaticians, the brine graphs are a mathematical concept which allows us to assemble genomes from short reads, and it's used by genome assemblers like Spades. And based on the quality of the data and the sequences themselves, the graphs can be more easy to interpret or more difficult to interpret. And they do not correspond to the physical structure of the DNA because the DNA is all shredded before sequencing. They only illustrate the sequence similarity based on which the assembler can determine what was the original genomic sequence. And even in simple cases like linear chromosome and circular plasmid, the graphs can quite quickly get quite complicated. So what did I observe? First of all, I was able to obtain mitochondrial genomes of 25 phototrophic species, which covers 14 of 50 euglenophyte genera, including some very mobile species like Euglena ehrenbergi or Euglena velata. And in all cases, I determined the structure and gene content. So this is how it looks for Euglena. In here you can see this assembly graph of Euglena hemalis. And we can clearly see those conserved, because they are well-behaving gene cassettes, which contain 1 or 2 genes. And it corresponds to the structure I described previously. And this is the most typical structure I observed across the tree. 22 of analyzed species look like that. But I was also able to identify 3 perfectly or almost perfectly circular genomes in 2 Colacium and 1 Eutreptia species. So Eutreptia is early branching marine species. And what is interesting about them is that they all encode the same 8 mitochondrial proteins. And when we look at the gene content of all of those, of all Euglenids, they more or less all have the same gene content. Those gaps are distributed rather patchy, and it may be due to technical issues. Maybe the sequencing run wasn't good enough, or maybe the gene itself is more divergent. It was difficult to identify it. The same story is with mitochondrial ribosomal RNA genes. It was relatively easy to identify them within the those taxa which are closer to Euglena gracilis, but more difficult to identify them in more divergent taxa, with notable exception of Eutreptia, because in circular genome there was no other place I could look for those RNA sequences, so it was easy to find them. And for the rest, I need to use more complicated methods. And last but not least is that I observed group 1 introns in several species, and notably those introns in COX-1 genes. They are all in the same positions, and they encode the same homing endonucleases, which allow group 1 introns to spread. So how do I interpret my results? First of all, when I started my research, there was only 1 mitochondrial genome of Euglena gracilis, and it wasn't really clear how did it come to this stage. In the meantime, a paper describing fragments of circular mitochondrial genomes in early branching Euglenids showed up, which suggested that common ancestor of all Euglenids had circular mitochondrial genome. And I can confirm those results with my own preliminary data coming from heterotrophs. For example, environmental isolate of Peranema has a perfectly circular mitochondrial genome, but I cannot tell you much more about it because this just came in last week. And if we also include other circular mitochondrial genomes I've assembled, it seems less clear. Like, I hope that with more data, I will be able to tell what happened during the evolution of Euglenids. But the more data I acquire, the less I know. And why do I know less? So looking at this tree, we can think that it's easy because this circular mitochondrial genome just reappeared in 2 places, in Eutreptia and in Colacium. However, there is no single described case like that organellar genome, which was circular, got fragmented, and then it went back to the full circle. And it's easy to explain that. It's a completely different thing for the cell or organelle to maintain the circular rather than linear genome. The cell requires different set of proteins, and if the ancestor of large group loses the circular genome, those proteins would go missing from the genome relatively quickly because there is no need for them without circular genome. So I went back to the graphs themselves. And as I told you, they are not always as easy to interpret as we could hope for. And while those graphs can be perfectly well interpreted as linear chromosomes with divergent ends, it's also possible that there is a larger circular molecule present in low copy number and a lot of short linear fragments. And if this was the case, there would be no need for regaining those circles in Eutreptia and Colacium. They would only just need to, for some reason, lose those linear fragments. The main question after I had this idea was, why would any organism do it like that? So I have few far-fetched answers to that. One is that the presence of group 1 introns suggests that there is, because group 1 introns are known for the impact on the plasticity of the genome. Like they allow the genome to shuffle itself more easily. So it would be one of the mechanisms how. But we know that in euglenoid nuclei, the transcription regulation is not working as well as we hope if we want to study some divergent transcriptomics, for example, differential transcriptomics, and that some genes which are required the higher level, which transcripts are required higher level, are present in higher copy numbers. So I can imagine that something similar is happening in euglenoid mitochondria, that those genes which are required in higher copy at higher level are replicated from this circular master copy onto linear chromosome, and those linear fragments serve just as the base for transcription to elevate the level of the transcript in the cell. Whether it's true or not, the facts I have for now are that there are definitely mitochondrial genomes, circular mitochondrial genomes in euglenoids, and it's not that clear if all the Euglenoids have linear fragmented genomes or not. Almost all Euglenoids have the same set of mitochondrial proteins within the genes, and so far we cannot really tell what did the mitochondrial genome of the ancestor of euglenoid look like. To answer, to remove this question mark from here, I plan to, in the second part of my grant, I plan to use large-scale cultures, fraction the cell to isolate only mitochondrial part, and sequence only mitochondrial DNA using long grids, and maybe confirm with some other molecular experiments. So that's all from me. I would very much thank the organizers for inviting me and all of you for listening to me. Thank you.
Thank you, Paweł. And we'll now continue with third speaker in this session, Metody Holender, who is a master's student also in the same Institute of Evolutionary Biology here in Warsaw at the University of Warsaw. So welcome, Metody, and he will be talking also on endosymbionts, but the bacterial endosymbionts of euglenoids.
Good evening, everyone. I will start sharing screen now. Once again, good evening, and I'm very glad to present my study on the Eutreptiella endosymbiont with use of metagenomic and fluorescence in situ hybridization analysis. I would like to start just to mention that the euglenoids, Euglenoids symbiosis are, as for now, as far as we know, poorly explored. We don't know much of them, and many of them were reported in the pre-molecular era. So we basically now cannot really compare it with the other data we collect. But one of the prominent examples here is the marine Eutreptiella, which was studied, and in 2013 it has its symbiosis interactions described, including the endosymbiotic bacteria. But this original research left some room for improvement as of now, because the back at the time it was the endosymbiont wasn't clearly identified, and basically we had not much information except for that it is inside the cell. So we had only the non-specific stainings, and the role of the endosymbiont was unknown. And it would be very interesting to actually to answer those questions, because it can be a very good model for symbiosis studies, since it's easily available for culturing. Therefore, we raised the questions: who is this endosymbiont exactly? Is it really single? How it interacts with the Euglenoids host, and how we can compare it to the other bacteria, its relatives, or other endosymbionts. Therefore, we designed this presented here pipeline of studies, where we would perform the Illumina sequencing on the DNA extracted from the Euglenoid culture. After that, perform the bioinformatic analysis in order to separate the genomes present in obtained data, and hopefully find the endosymbiotic genome itself. And then, as for the full 16S cycle, design a species-specific 16S probe and perform the fluorescence in situ hybridization for the final confirmation, as well from the obtained proteomic dataset of the predicted proteins to predict also some of its metabolic capabilities. In the sequencing, we've obtained the clearly identifiable endosymbiont, as well as low-quality assemblies of the ectosymbionts, also described in the original paper, but due to their low quality, not analyzed here. The endosymbiont's genome showed all the features in agreement with the known Rickettsiales symbionts, so the low GC content and the genome size reduction, and the obtained genome was of high completeness and low enough contamination. On the basis of the 16S RNA found in the assembly, predicted from the assembly, we performed a phylogenetic analysis, which yielded us tree of the family Midichloriaceae with the Euglenoid symbiont in a well-supported clade containing the previously described endosymbionts known as the genus Candidatus Grellia, including also in this branch, the closest relatives of Euglenoid symbionts, also the previously described endosymbiont of the Trichoplax, so the placozoan animal. And therefore, we concluded that it's likely belongs to the same species, so the Candidatus Grellia incantans. We therefore designed a monolabeled Grellia incantans specific probe and validated it on the available culture. Here you can see the results of those experiments. As you can see, we only the intracellular bacteria, so contained in the cells, are stained with this probe. And therefore, we concluded that yes, this endosymbiont is really single in that case. Also, from the analysis of the predicted proteins, we've inferred that the endosymbiont possesses actually highly complete, highly conserved core energy metabolic pathways, so including the Krebs cycle and the electron transport chain. Its biosynthetic capabilities were highly reduced, but we still found some simpler pathways for the amino acid synthesis and interconversion. as well as the systems thought to be a hallmark for the maintenance of the symbiosis, such as type 4 secretion system, thought to be able to deliver effector proteins directly into the host cytoplasm, as well as the ATP-ADP translocase, thought to take part in the energy parasitism. We've also found the enzymes for synthesis of the cell wall and the lipopolysaccharides, as well as the pathways leading to synthesis of a number, actually, of the coenzymes, such as vitamins B6, B7, riboflavin, FAD, and coenzyme A. From that, we've answered all of our initial questions. We've confirmed that this environment belongs to the Candidatus grellia, and from its predicted metabolic capabilities, we think that it can be at least partially energetically independent, so able to synthesize its own energy from the biomass provided by the host and not take the ATP directly, at least not always. Found type 4 secretion system suggests active manipulation with the host cell biology, but as for now, we were not able to clearly point at the effectors for this interaction. Still, despite some preserved synthetic pathways, it still is almost fully incapable of synthesizing a lot of the crucial compounds and relies on the host for important metabolites, such as the diversity of the amino acids and also other molecules such as nucleotides. However, we think that it may provide at least some coenzymes for the host, which was the one of the mechanisms also proposed for the previously found grellia incantans in the trichoplax, where it was hypothesized that it can synthesize the riboflavin for the trichoplax. Therefore, on the basis of those features, we can conclude that it still is very similar to the to other closely related bacteria, such as from same genus Grellia and Midichloriaceae overall, which suggests one of the most probable lifestyle for them is to be not strictly a parasite, but rather an opportunistic commensal, so able to invite a variety of hosts, but without directly actually harming them. However, our study was, as for now, limited, mostly because we weren't able to have a look into the transcriptional capabilities of this endosymbiont. And therefore, in the future studies, we plan to first improve quality of the genome, endosymbiont's genome assembly via the nanopore sequencing, as well as to have a look on what its actual capabilities are via the transcriptome sequencing, because it will tell us a lot more regarding whether it is really capable of producing, for example, those remaining amino acids from the remaining pathways, or it is not the case, and also maybe what are the effectors for the interaction. Also, this is on our established bioinformatic protocol and some preliminary studies we've done on the single cell analysis. We plan to expand those analyses, so endosymbiont hunt on other protists and specifically other euglenoids to expand our knowledge in that topic. Thank you very much, and I would like to thank you, my team and National Science Center for funding this research. Thank you very much.
Thank you, Metody. And with that, we close presentations for this session, and we're open for the discussion. And I will start asking one question myself, and then we'll go with the questions which are already piling up in the chat. So I have a question for Vladia, since so actually I was wondering, I really like this concept of combining some types of I don't know, trophic stages, let's say, and analyzing this gene content in these groups. But I was wondering in Euglenoids how that you put together actually all the heterotrophs together, the osmotrophs and then the, I don't know, bacteriivores and eukaryotes. And I would expect that the income of the foreign genes would relate, maybe will be related with actually the eukaryote, right? So I don't know, this is more like a question versus how do you interpret this? What is the, how they get these genes if they're osmotrophs, basically?
Yeah, thank you for the question. I would like to apologize that I was too long at the first because I didn't see the chat warning popping out. But to your question, I understand that it is well, if we had a denser sampling, I think the fairest way would be to group the phylogenetic the clades, you know, to have like 3 representative of individual clades, and then if we have like phylogenetically unified groups, that would be better. However, we didn't have such a dense sampling, so basically the idea was to group the taxa before that branched out before or after the putative endosymbiotic event, which was at the common ancestor of Euglenoids. So we grouped like heterotrophs that branched off before and autotrophs that branched after, with the exception of Euglena longa, which lost the photosynthesis. So we thought that, yeah, obviously it's a special case, so that would bring undesired variants into our analysis, but we put it aside.
And maybe like a small addition to this question. So in these genes which were acquired in different, because you mentioned that they were pretty much species specific, so the transfers. So did you see the patterns, like the same genes were transferred, or you didn't look at that? Like what kind of genes were actually transferred besides the origin?
Oh yeah, so that's the phase where we are heading, like to annotate which types of genes have been transferred when. No, so no, I don't have answer to this question.
But it will come, so we are looking forward. Okay, so I'm now heading to the chat. I see questions. So Alistair Simpson asked to ask a question in person because it's too difficult to type in, and it will be a question for you, Vlada. So Alistair, if you're there.
Can you hear me?
Yes.
Yeah, mine, it's actually pretty similar to Anya's question. I was sort of wondering if there's a way of looking at your data, particularly I'm thinking particularly from the heterotrophs, and sort of testing model, like having an explicit test of the model where compare the plausibility basically of the genes of, let's say, haptophyte origin coming from a single endosymbiosis, sorry, a single contact. So let's say an endosymbiosis versus acquisition over time, like whether that's something that, is there some sort of framework in which you can test those alternatives as models against your data? I guess my intuition, I think, is the same as yours, that probably those genes from, say, haptophyte origin or ocrophyte origin or whatever have been coming in gradually through individual lateral gene transfer events, perhaps from prey. But is there a way that can be sort of, if the distributions of genes can be tested, given that you also expect things like loss and so on, if there was an endosymbiosis?
Yeah, thank you. So again, for that, we would need denser sampling of that data, like much denser data set, and maybe then we would see a pattern. But I'm worried that even with with our sampling in the last or like the previous last slide with that tree where we were positioning this like incoming gene events on that tree, we basically haven't observed any trend besides the one that I noticed that I noted that there is increased green influx in the common ancestor of Euglenoids. Other than that, it was, and that was a little disappointing, super noisy. So I'm afraid that even with the denser sampling, my expectation is that we would not see any good trend, probably because, yeah, it's coming gradually, and the reconstructions are too prone of errors, and so it's difficult to disentangle any trend. That's my expectation. Maybe I'm too pessimistic, but yeah.
Who knows? I completely agreed on the denser sampling being important. Thanks.
Thank you.
Okay, now I'm going to the next question, which is coming from Vlad to Pavel. So I will read it. Pavel, you mentioned that cell needs a different set of proteins to handle circular mitochondrial genome than linear. Do you see proteins for maintenance of the circular mitochondrial DNA in the euglenic dataset?
Yeah, so for like those proteins would definitely be encoded in the nucleus, and we have only checked so far the Euglena gracile schemalis longa transcriptomes because they are relatively high quality. And in those, there are proteins that might or might not be those helicases and topoisomerase. Like I don't remember all of them right now. But I'm waiting with checking them for some more transcriptomic data for species like Colatium, which have definitely circular genome, and then I want to compare like those who have those weird genomes with those which have circular. If there is some pattern, it may be easier to identify those critical proteins. I hope that answers your question.
And next question to Pavel from Erin Morrison. What would be the advantage of a linear versus a circular mitochondrial genome?
So if we speak about completely linear genome, like genome composed of multiple linear chromosomes, I have no idea. I cannot imagine like clear advantage of such genome composition, and also in kinetoplastids and diplonemids. Though those mitochondrial genomes can be 250 megabase pairs large, so it's like 1/12 of human genome, and they encode 4-6 proteins. So I cannot really imagine any advantage in that, but it may be just a case of neutral evolution. So basically, even if those euglenids have those linear, truly linear mitochondrial genomes, they just didn't die yet. Like it's not bad for them enough to actually kill them. I don't know if anyone has any idea how it could be beneficial to the cell. I'm open for ideas because I cannot think of any.
Okay, and the third question also to Pavel from Gordon Lex. Have you tried automatic organolar genome assemblers and how they perform in comparison to your manual approach?
Yeah, so I definitely tried them. I wasn't happy with the results, but right now I don't remember why exactly. I would have to check my data, but I definitely tried them in species which were far from Colatium, so like Euglena, and maybe in those species which are closer to Colatium, where those mitochondrial genome assemblies look more clear. Maybe it will actually be possible to obtain the whole circular sequence with novoplastid get organelle. I will definitely try that. Thank you.
Okay, it seems like Gordon is happy about this answer. And now the question to Methode from Gladia. What is the advantage of the symbiont to encode ATP-ADP transporter if it has fully developed energy metabolism? And does it have also sugar transporters in the membrane?
Yes, so actually, it is thought that the presence of the ATP-ADP transporter may be an addition to its own energy metabolism. So one of the hypotheses is that it can actually at one point just generate the energy by its own, or to tap into the host's energy pool. The other is that, but it's I would say it's less confirmed. We don't have, as far as I know, any proofs of that. Is that actually the main energy uptake can be from the translocase, and the machinery left from the energy metabolism may be repurposed for other things, like, for example, in case of ATP synthase to create the proton gradient. But actually, I find it not so likely, due to the fact that it has not only, for example, the ATP synthase, but all the pathways from the Krebs cycle beginning and ending on the electron transport chain. Regarding the sugar transporters, we didn't found the phosphorylase system. So, but in that part, our analysis may be lacking for that in terms of the transporters overall, because the primary tool we've used, the KEGG database, doesn't seem to be of the best results here.
Thank you, Methode. And I think last question maybe in this session from Natalia Gumińska to Paweł. What is the advantage of manual approach again, based on assembly graphs instead of the pipeline dedicated to investigation of structural variants?
Yeah, so to clarify, do you mean pipelines which require reference genome, or those which work de novo? Because those which require reference genome, of course, we cannot use for Euglenoids. And I've tried several, I would check exactly which, but definitely like plasmid space was in there.
No, Paweł, I don't mean the plasmid space and other assembly methods for the entire genome. I mean the tool sets for instance, for circular particles like Circle Map, Circle Explorer, and Grab.
I don't exactly remember which of these tools works based on the genome assembly like space and which requires better reference. And I definitely tried the last two of them, and I wasn't happy with the results for some reason. But right now, I don't think that it was pretty long time ago. But one of the main advantages I can tell you right now is that in several cases, there were some proteins which I first missed using my blast searches, because they were divergent enough that it was difficult to tell if they are euglenoid mitochondrial protein or alpha protobacterial contaminant. But when I saw on the assembly graph on my own that they are all clamping together, I knew that even if it's not really certain whether it's like on the sequence level, it's not certain if it's mitochondrial protein, but because it's clamped with other certain mitochondrial proteins, I knew that this will be mitochondrial protein.
Thank you very much for your answer.
Thank you, Paweł. And well, I think we have 2 more minutes, so maybe we can go with the really last question. Alistair Simpson again, now asking Methode, has there been any attempt to systematically survey Euglenoids culture collection for bacterial symbionts?
So as far as I know, there was no single study which would take this task, but also a majority of the Euglenoids genera were never observed so to harbor the symbionts. And therefore, we actually, as for now, we want to do such survey on the cultures we have access to. So in order to clarify and to expand our understanding on this topic. Therefore, we try to approach this from the metagenomic point of view, whether we can or cannot find any hints onto the endosymbionts in the metagenomic data from the cultures.
Yes, maybe I will comment on that just very briefly. It's basically the data set we have, what Pavel has, or my previous PhD student Kasper on plastids. Obviously, we can test if they have any possible endosymbionts. So this is what Method is doing now. So there have been known, but right now we are checking that. And I see this is the end of this session, and I'm giving the voice to the organizers.
Thank you very much, everyone. Excellent presentations this morning. Special thanks to Vlada, Pavel, and Metody, and of course, Anya for her assistance with the chairing of the session. So we'll take a 15-minute break now, and we'll come back at half past the hour. And we'll start with session 2. Thanks again. See you soon.
Thanks. Scott.
Yes.
Hey,
Alistair.
hey. So just to check, sorry everyone else for yakking over the top of this. So Ria Harada is giving, you have a video for him?
I do, yes, I'll present that, no problem.
Right, so there's nothing I need to do to, brilliant.
Yep, I'll broadcast that, or my colleague will broadcast it over the laptop, and we'll just allow everybody to watch that and view that.
All right, and I wasn't clear on, sorry for this, but I wasn't clear on the screen sharing and unsharing. That's people are managing to do that themselves, right?
You got it, yep.
Brilliant, thank you.
Is that it?
I think so.
Okay, awesome. We'll see you in
I've got I'll have the interesting task of keeping Marek Elias to time, but
okay. All right, we'll see you in a few minutes.
All right, cheers.
Thanks.
Hi guys. I will try my best, to.
I'm just teasing. I'm just teasing.
Yeah, but honestly, I may be late. Yeah, so. Okay.
All right, thanks. Okay, session 2, welcome back. I think I'll just get started in introducing our session moderator, Dr. Alistair Simpson. Dr. Simpson is in the Department of Biology at Dalhousie University on the east coast of Canada. So hello to a fellow Canadian researcher. Alistair's research focuses on particularly overlooked protozoa and what they reveal about the evolutionary history of eukaryotic cells. Dr. Simpson also examines the diversity of free-living protozoa in unusual environments, so we're a fan of that, as well as ecologically important forms from sediments. His recent work has looked at the detection and characterization of symbiotic or parasitic protozoa of marine invertebrates and algae. So with that, I'll hand it over to you, Alistair, and look forward to the session.
Thank you very much, Scott. Our first speaker in this session, where we're continuing our discussion of euglenoid evolution, is Marek Eliáš from Ostrava in the Czech Republic, talking on the evolutionary cell biology of plastids. So please, Marek, if you could take it away.
Okay, so hello everybody. Can you hear me?
Yep.
Yeah, and can you see the slide? The presentation? Okay, good. Yeah. So hello once again, and thank you very much for inviting me to present at this meeting. It's my first participation at this Euglenoid meeting, and I'm very excited to have an opportunity to present stuff that I did not simply, or I could not present in the recent years because of the COVID restrictions. So I will tell you some stories that are already published, coming from my lab or from work that we have done in collaboration with a number of people whom I will thank right away because I may not have time at the end of the talk. So above all, people from Prague, Vláďa Hampl and his former student Anna, the lab of Mirek Oborník from České Budějovice, especially Zoltán Fishy, and Ania Karnkovská, Rafał Milanovský, and also Štěpánka from Brno. And the 3 people shown with the photo in the right corner, those are those that have contributed most to what I'm going to tell you. So basically, I'm going to speak about the plastid in euglenophytes, and I'm grateful to Vláďa for providing a very useful and helpful introduction into the evolutionary origin of the plastid in euglenophytes. So we have heard that it's derived from Pyramimoda dalian alga, and basically what I want to show is that we should simply stop thinking about the euglenophyte plastid as a simply green algal plastid or a chloroplast, because there have been a lot of changes that have remodeled or transmogrified the original chloroplast into something that is very far from it. And to think about these changes, I use a very simple framework. Basically, I think in terms of losses, replacements, and gains of individual components of the plastid. So let me illustrate these type of changes with specific examples. And I start with one thing that is rather peculiar. So when analyzing the data that we have generated for Euglena longa and that had been available already for E. gracilis, we were surprised to find out that we cannot find any homologs of known components of the plastid import machinery, or particularly the complex that resides in the outer chloroplast membrane called the TOC complex, which led us to propose, and it's still unproven, and I think it's a matter of debate, whether what was the nature and origin of the individual membranes that make an envelope of the plastid of the euglenophyte plastid, and based on the absence of the TOC complex or of its loss, we would also hypothesize that it's the outer membrane as such that has been lost during the integration of the green alga plastid into the euglenophyte host cell. There are other components, I'll say housekeeping components of the euglenophyte plastid that are surprisingly missing. For example, the signal recognition particle over here, which is an important part of target mechanisms targeting proteins into the thylakoid membrane, and which is essentially omnipresent in at least photosynthetic plastids, yet surprisingly missing in euglenophytes, just suggesting some simplification of the targeting processes. There are also obvious departures from the standard mechanism of plastid division machinery, because there are no homologs of the dynamine ARC5 implicated in plastid division in basically most other plastid-bearing eukaryotes, and even more surprisingly, there is no FTSZ, that ancient bacterial or cyanobacterial component that also is a crucial part of the plastid division machinery, making the mechanism of euglenophyte plastid division presently completely unknown. It's not only the housekeeping processes, but also the, let's say, the metabolic pathways that are the very reason why the plastid is kept by the cell that may be surprisingly missing. So based on the reconstruction of the plastid of Euglena gracilis plastid, which was facilitated by a combination of bioinformatic analyses of available transcriptome assemblies and proteomic data obtained by a group of labs led by Vlaďa Hampel. So based on this, we surprisingly concluded that there is essentially no role of the Euglena plastid in amino acid biosynthesis, which contrasts starkly with what's normally thought of about plastids in other groups, where usually multiple amino acids are synthesized, and the plastid is typically even the sole source of the particular amino acid for the whole cell, like, for example, the aromatic amino acids tryptophan, phenylalanine, tyrosine. So this is really something rather unusual. In addition to complete pathways, there are also some surprising so-called pathway holes. So individual enzymes that are expected to be present to make a pathway complete, yet missing, missing in that no homologs of the known enzymes of the known proteins securing the respective catalytic role identifiable in the data. I will just highlight two such examples. One is an enzyme of the sulfolipid biosynthesis, which is responsible for the synthesis of UDP sulfokinase, which is again omnipresent not only in plastids, particularly photosynthetic plastids, but also it's present in cyanobacteria and so on. Yet no homolog can be discerned in any euglenophyte for which we have appropriate data available, making it completely unclear how this compound, which is then used and apparently by an enzyme that is present to make sulfokinase glycerol. So how this compound is made, it's completely unclear, and it points to a potentially completely novel biochemistry that Euglena has adopted for this task. Another interesting, really unique absence or gap is the lack of this enzyme, the so-called geranyl phosphate reductase, which again is common in essentially all photosynthetic eukaryotes and also in prokaryotes, yet no homolog of this enzyme can be found again in all euglenophytes. It's in the end, it's not that surprising because previous biochemical analyses, I believe in 1980s, established that this phytyl pyrophosphate, which is the product of the reaction catalyzed by this enzyme, is in fact not made by the common plastid isoprenoid biosynthetic pathway, and the precursor comes from a cytoplasmic source of isoprenoids, meaning that there must be a completely alternative pathway, and we believe it includes free phytol as one of the intermediates. Yet we have absolutely no idea how phytol itself is made. So again, this points to potentially novel enzymes and interesting biochemistry going on. Okay, when speaking about loss in euglenophyte plastid evolution, there are extreme examples in the non-photosynthetic euglenophytes. I mean, those that are secondarily non-photosynthetic, and one of them, and the best studied, is Euglena longa, which is one of my pet organisms, and this is something that we are studying quite extensively. It was actually, or it had been, in fact, unclear whether plastid is present at all, and this has been resolved in 2000 when a complete plastid genome sequence was obtained from this species. So obviously the plastid organelle as such must be present. Interestingly, the only gene in this plastid genome that does not code for a housekeeping component that itself is involved in genome expression or genome maintenance is the RBCL, so the gene for the large Rubisco subunit. As you can see, in fact, the sequence is extremely divergent. You see this long branch suggesting a highly accelerated evolutionary rate, and yet still, this is apparently the reason data for the retention of the plastid genome. And of course, this raises the question: what's the function of the Rubisco enzyme in a non-photosynthetic plastid? To answer this, or in attempt to answer this, we have sequenced the transcriptome of Euglena longa, I would say a high quality transcriptome, and explored it to reconstruct the metabolic pathway of the Euglena longa plastid. And we have found a couple of pathways or metabolic modules most likely present, which this in the plastid. This was highly facilitated by the availability of proteomic data from Euglena gracilis, so we could make a nice comparison. So I will now present a few figures comparing Euglena longa and Euglena gracilis, so the reconstructed metabolic maps for both species. So one surprising finding that we made was that Euglena longa, despite being non-photosynthetic, has preserved biosynthetic pathways for tocopherol and phylloquinone synthesis, which are compounds that are, so these are the pathways here. These are compounds that are typically associated functionally, or at least thought to be functionally associated with the photosynthetic functions themselves, or with the photosynthetic processes. Yet, apparently, they must have a function independent on photosynthesis. We were able to confirm their presence in E. longa experimentally, so it's not just a bioinformatic result. We may possibly think about their role in redox regulation or protection against oxidative stress or whatever. Concerning fatty acid or lipid metabolism, which is common in plastids, fatty acid biosynthesis as such, which is a common function of plastids, is missing in E. longa. Yet it still synthesizes certain types of lipids, phosphatidyldiacylglycerol, galactolipids, and sulfokinoacyl diacylglycerol that I mentioned before. This is not that uncommon. This is not without precedent in non-photosynthetic plastids. Yet there is something very interesting concerning sulfokinoacyl diacylglycerol synthesis in euglenophytes. The first thing is the absence of the enzyme that makes sulfokinoacyl UDP. I have already mentioned that. There is another thing, actually two different types of the second enzyme that makes the SQDG itself, the so-called eukaryotic SQD2 eukaryotic type, and the bacterial type SQDX. They are related, yet distinctly different. And interestingly enough, while Euglena gracilis has the standard expected SQD2, it also has this bacterial SQDX, and it shares it with Euglena longa, for which this is the sole representative of this enzyme. And this is the first case of the presence of this bacterial type enzyme in any eukaryote. Of course, raising the question, what's the reason for having two different enzymes for the same reaction in Euglena gracilis? And in fact, its presence as the sole, the presence of SQDX as the sole enzyme in Euglena longa, together with experimental evidence for the presence of sulfokinovosyl diacylglycerol in the species, indicates that this newly acquired enzyme must be evolved or must have the function in SQDG synthesis, because there is no other option how to explain the presence of this compound in E. longa. Okay, of course, what is most interesting probably is the RBCL, the question of the function of the RBCL, which is linked to the Calvin-Benson cycle. And interestingly enough, E. longa has most enzyme of the Calvin cycle except two. So these two enzymes here are missing. So in fact, instead of a cycle, we speak about a Calvin-Benson pathway, a linear pathway. Now the question is, what's the function of this pathway? A potential explanation, very important clue for this is the conservation of a mechanism, or at least the components of a regulatory mechanism that regulates the functioning of the Calvin cycle, depending on the redox status of photosynthetic plastid related to the functioning of the photosynthetic machinery itself. And this is surprisingly conserved in E. longa, and we believe this actually provides a clue for the explanation for the retention of the Calvin-Benson cycle. Without going into detail, we propose that it serves as a kind of redox valve, that it helps to regulate redox balance through because it can consume extra electrons basically by fixing CO2, so it can help to keep the redox balance in the cell. And we can discuss it, or we may discuss it at the end if you wish. And the last thing that I want to say about E. longa is that based on the data that we have, it seems that there might be a remnant of the heme biosynthetic pathway in the plastid. Interestingly, E. gracilis has two parallel pathways. One is mitochondrial cytosolic, one is plastidial. The mitochondrial cytosolic is present in E. longa, together with, let's say, enzymes that we could find in the data corresponding to the plastid pathway. However, the first enzyme, this aminolevulinic acid dehydratase, when inspected in detail, bears mutations at critical, catalytically critical positions, indicating it's not an active enzyme. It has been not pseudogenized, but pseudoenzymized, let's say. So it's a pseudoenzyme and serving probably some regulatory role. Again, we have no idea what is it. So what's the role then of this uros, which presumably makes uroporphyrinogen in the plastid? And the possible explanation is that there is actually a second branch, second metabolic branch starting from uroporphyrinogen, leading to cyroheme. It's shown here. We then looked for homologs of the enzymes of this pathway, and indeed we could find them in both E. longa and in gracilis, with potential evidence that they are localized to the plastid. This has not been confirmed experimentally yet. Interestingly enough, there is no known eukaryotic type of this enzyme. Even though cyroheme is synthesized even in plants, the identity of this middle enzyme, precorin 2 dehydrogenase, has not been established at the molecular level. So in this context, it's interesting to mention that the homolog of this third enzyme of the pathway in euglenophytes is fused as a translational fusion to a member of a family of uncharacterized reductases, which actually, when you check their distribution in eukaryotes, perfectly match the distribution of the other enzymes of the pathway. So we hypothesize that this is basically the missing precorin 2 dehydrogenase in eukaryotes. So curiously, by studying Euglena longa, we can potentially inform biology of photosynthetic algae and even plants. Okay, so in addition to losses, there are also, of course, replacements, like the one-to-one replacements from different sources. Vlade Hampel has spoken about this extensively, so I don't need to discuss this at all. In addition, but what I would like to stress are bacterial sources of euglenophyte plastid proteins. The role of bacterial genes in the establishment of primary plastids has been studied quite extensively, yet very little attention has been drawn to the role of bacterial genes in the establishment of secondary plastids. So in this context, again, it's interesting to mention that indeed there are euglenophyte, like specific euglenophyte plastids acquired from bacteria, like for example, sulfide reductase, replacing the original cyanobacteria-derived sulfide reductase in other algae. Yet even more interesting examples are replacements from host cell, let's say, source, direct, at least potentially coming directly from the eukaryotic host cell. And an interesting example is ribosomal protein L24, which normally in plastids comes from the cyanobacterial source, so it has been inherited from the cyanobacterial ancestor. Yet in euglenophytes, we could not find it, and instead we found a second copy resembling the eukaryotic distant homologue called L26. It's a member of the same older family. At the sequence level, it's very, very different. And this second copy, present solely in euglenophytes, is endowed with a plastid targeting pre-sequence. So we believe that it has replaced the original bacterial L24 proteins, resulting in a kind of chimeric eukaryote or cyanobacterial eukaryotic ribosome in euglenophytes. I could not resist mentioning that later we found out that there's another eukaryotic group that independently has made the same replacement. It's another group of my interest, eustigmatophyte algae, that are distantly related to euglenophytes, and they have done the same replacement. So there is a kind of preponderance for this exchange. Okay, so now finally gains, and this will make me busy till the end of the talk. So yeah, I could speak quite a lot about gains. I could mention, of course, this diversity of introns gained by euglenophyte plastids and the associated splicing machinery. There is a very interesting case that we have discovered of a second subsystem acquired from a chlamydial source. I should mention that plastids are normally endowed with a cyanobacteria-derived subsystem for FS cluster assembly, yet euglenophytes have apparently another parallelly functioning subsystem of a chlamydial origin. We don't have a really good explanation why there is such a redundancy. Another type of a gain in euglenophyte plastid evolution is the tendency to import proteins or fused proteins that include multiple protein units that are processed by still uncharacterized protease upon transfer into the plastid. This was originally described in 1980s and 90s, and for Rubisco small subunit and light harvesting complex proteins, which are imported as polyproteins, including multiple units that are then processed into individual proteins proteolytically. And it was in this context, it was interesting that we have discovered a series of proteins that are, again, translational fusions. In this case, typically different proteins that are presumably. So this is the N termini correspond to the plastid targeting pre-sequences, and these are these fused proteins that are presumably processed potentially by the same unknown newly acquired machinery to liberate the individual proteins. Okay, and the rest of my talk will be devoted to something that is the most recent work in my lab, and that deals with, or that relates to a curious discovery of a complex molecular machinery taking care, let's say, of 3' ends of plastidyl RNA molecules. And everything started a couple of years ago when we reported on the presence of polyadenylation of ribosomal RNA, of RNA in plastids of Euglena gracilis. This was the first report of such a phenomenon in secondary plastids at all. So now with all the new data and resources, we want to understand this phenomenon in more detail by searching the available transcriptome data from E. gracilis, E. longa, and so on. We found out candidates for enzymes responsible for the synthesis of these RNA tails. So one, this PNPase, is not that surprising because it's common in plastids. Yet, in addition to this, we found four more proteins that are totally unknown from plastids, yet there is evidence basically from the structure of the proteins themselves, and in 3 of them also for proteomics that are indeed localized to the plastid, and that may be involved in the synthesis of these tails. So these are examples of recruitment of, let's say, cytoplasmic or even nuclear proteins into the Euglena plastid, and this is completely unprecedented. And so we have taken an approach to directly modify or directly edit the Euglena gracilis genome by CRISPR-Cas system, which has been recently established by Nomura and collaborators to probe for the function of these enzymes and to test their link to the poly-A tail synthesis. And thanks to the effort, particularly of my postdoc Irka Pergner, we could get knockout, which interestingly enough have phenotype related to the plastid. They are it's apparent on the pigmentation, and this concerns the TNT2 gene as well as the PNT1 gene. So apparently the plastid function is disrupted. These mutants have no measurable oxygen production, and the quantum yield of photosystem 2 is 0, suggesting that there is an indirect impact of mutating or knocking out of these genes onto the functioning of the plastid that that is such severe that even the photosynthetic function is disrupted. Basically, proving that these proteins or genes are critical for the plastid functioning. And more recently, we have managed to obtain mutants of the other 3 genes, including the PNPase, where the phenotype is even more severe compared to the wild type, compared to the TNT2, for example, mutant. While the knockout mutants of the 2 other proteins or genes are, as far as we can say, don't have such a profound phenotypic consequence at the pigment level. So now, of course, the challenge is to show what's the real or what's the most immediate impact of these knockouts. So we are now going to study the structure of the RNA tails to show directly the involvement of these proteins in the RNA metabolism of euglenophyte plastids. So to conclude, basically, you can read. I don't think I really need to read this. So basically, I wanted to show you on specific examples how complex the integration of the chloroplast, from Peramimodala alga into the ancestor of euglenophite host was. How many changes have been employed, and how far the euglenophite plastid is from the green algal paradigm. And I think we are approaching exciting times with all the tools and resources available. We can spend the rest of our lives just to understand the biology of euglenophite plastids. So yeah, that's it. Thank you very much.
Thank you very much, Marek, for incredibly wide-ranging and interesting talk. So please, if you have any questions for Marek, we'll ask them after the next two talks, but please feel free to place them in the chat so we can get to them in due course after the next two talks, as I said. The next speaker we have is Dr. Gordon Lax from the University of British Columbia, who will be talking about the phylogenetics of phagotrophic euglenoids.
Thank you, Alistair. I assume you can hear me and see my screen?
Yes.
Awesome. Well, thank you, Alistair. So we've heard a lot about phototrophic euglenoids, obviously, over the last few days, but we've only started to hear about phagotrophic euglenoids today in a couple of talks today. So I would like to focus a bit more on that and just talk about, their phylogenetic diversity of these phagotrophic euglenoids. And I should say this is work that I've been doing in the Keeling lab at UBC in Vancouver in collaboration with Anna Cho. So we frankly know quite a bit about phototrophic euglenoids if you really think about it. We know a lot about their biodiversity. We know a lot about their evolutionary relationships. We do know a lot about their taxonomy, and we do know a lot about their biotechnological potential. But compare this to phagotrophic euglenoids, where we don't even really know how many species there are, or even how they're related to each other. Don't even speak about ecology or biotechnological potential. And this is despite phagotrophic euglenoids likely stemming the vast majority of the phylogenetic diversity of euglenoids. And if I say phagotrophic euglenoid, I mean a euglenoid that likely never acquired the ability to do phototrophy, but rather feeds on bacteria and or other single-celled eukaryotes. So why should you care about phagotrophic euglenoids? I feel like that's a good question to ask. Well, it turns out, as we just heard, particularly in Marek's and Vladia's talks, that phototrophic euglenoids arose from within phagotrophic euglenoids through a secondary endosymbiotic event with a Paramimmonadelian green alga. But we don't really know what the phagotrophic host in this interaction looked like. But if you look at this cartoon version of recent SSU trees or recent SSU phylogenies, you can see that we do have phototrophic euglenoids as a monophyletic clade up here, and pretty much everything else that you see is a phagotrophic euglenoid genus. Some of these genus names are in red, and for those we only very recently were able to acquire molecular data for the very first time. So prior to that, no molecular data existed whatsoever. If we look at SSU trees or SSU phylogenies, we can actually see an interesting trend here, and you can probably already figure it out. There is this big group up here called Spirocheta, and that is something that we recover in all of our phylogenies nowadays. And it kind of makes sense from a morphological standpoint as well. So this clade of Spirocheta includes phototrophic euglenoids, which again includes things like Euglena, but also a whole bunch of other phototrophic euglenoids, but also like a pretty big diversity of phagotrophic euglenoids. All of these taxa have a certain number of pellicle strips. So pellicle strips are these proteinaceous strips that run underneath the cell membrane in all euglenoids. And it turns out cells or taxa within Spirocheta tend to have many pellicle strips, sometimes over 100. And if you think about Euglena, they can undergo this euglenoid metaboly thing. That is only possible because of a large number of pellicle strips. Turns out, as soon as you go outside Spirocheta, so if you go closer to the base of the euglenoid tree, you only find phagotrophic euglenoids. And these phagotrophic euglenoids tend or only have between 4 to 12 pellicle strips, not always very rigid. And there's an SEM down here of a Pedalomonas that has 6 pellicle strips. So these are very rigid cells. What you can also see here is that the backbone in this tree is completely collapsed, and that's just because we don't know what the relationships look like when we just use the SSU gene to estimate our phylogenies. So one way around this is to, well, not use the SSU gene only, but use rather, a larger number of genes, do a multi-gene analysis or a phylogenomic analysis. And the traditional way to do this would be to have your organism of interest, bring it into culture, and then sequence the transcriptome, and then pull out your genes of interest from the transcriptome and do phylogeny. The issue is, as several people have touched on today, phagotrophic euglenoids tend to be, a bit tricky to culture because they need prey, and sometimes they have very specific prey preferences. And to figure out what actually is culturable or bring it into culture can be very time and cost intensive, actually. So something that I've been doing during my PhD with Alistair, actually, and during my time as a postdoc here at UBC, is actually do single cell transcriptomics, and chiefly a method called SmartSeq2. So with this, basically, we isolate our cells of interest from the environment. We photo document them under the microscope, or sometimes video. And then we basically put the cell via manual isolation into a tube and do the SmartSeq2 protocol. It has an mRNA selection step, and then the reverse transcribed cDNA gets then sequenced on an Illumina platform. And in the end, you hopefully have a pretty decent single cell transcriptome, out of which you can then pull your genes, your marker genes of interest, and construct a phylogeny. And this is a tree from a paper that we published last year. This is the first, as far as we know, the first multi-gene analysis of euglenoids to actually include a reasonable amount of phagotrophic euglenoids. I'm not going to spend too much time on this. This is a 20-gene tree. I cut off the outgroups because they're not interesting right now. But we do find euglenoids as a monophyletic clade up here, so all those colored boxes. And then we have sister to them, kinetoplastids, diplonemids, and symbiontids. Again, within euglenoids, we can see this clade of Spirocheta up here, which again includes phototrophy euglenoids, but a whole bunch of other phagotrophy euglenoids. And then at the very base of the euglenoids, we can see a group called Pedalamonadida, or pedalamonads, in orange here. And these, in our phylogenies, appear to be the deepest branching euglenoids that we currently know of, at least. And kind of in between Spirocheta and Pedalamonadida, we have this loose assemblage of cells called ploecids. And these ploecids share certain morphological characteristics. They always have 10 to 12 pellicle strips, and they do have two flagella. They always glide. They always glide on their posterior flagellum, and they can be either bacteriovorous or eukaryovorous. Now, despite sharing certain, morphological similarities, it turns out these taxa are actually not particularly closely related to each other, but rather fall into, in this tree, 4 different clades, and the exact relationships to these clades is not entirely clear. You can see I call it the mid backbone of the tree is not particularly well resolved here. So to me, this kind of screams, well, maybe there's a bit of diversity missing, that we haven't sampled yet and have not been able to put into the tree. And potentially, if we find more ploecid taxa and sequence them and put them in the tree, we would resolve this uncertainty at the mid backbone of the tree. So turns out, this is a video of one of the ploecids, just to show you a little bit. So this is an Alcasia. You can see, if you look closely, you can see the pellicle strips running through it, and you can see it has 2 flagella. You'll see the video later again, so you can look at it again. So it turns out Anna and myself, over the last few months, were actually pretty successful in finding additional euglenoid taxa in the environment. Turns out we isolated 5 new cells and got 5 single-cell transcriptomes. 3 of those fall into, we could actually identify down to genus and species level, 2 of which belong to the genus Hemiolia. So you can see one here and one here. And then another one to Decastava. And for these, we had SSU data prior, but now we actually have multi-gene data for the first time ever. So it's actually kind of pretty exciting to have multi-gene data for these guys as well. What's even more exciting is that we have 2 of the 2 remaining cells that actually appear to be novel taxa, or at least we made up new genus names for them. So these we actually found in a paper a while ago, a couple of years ago. But we could only identify them as like unidentified ploecid because there was no description that we could find. Basically, these seem to be novel organisms. So we had SSU data for these, but now we found very closely related organisms. So we have additional SSU data, but we now also have multi-gene data for these taxa. And because their position in the tree in the SSU was prior very unstable and they were jumping around a lot, it's very exciting to have multi-gene data for these taxa now. And hopefully this enables us to place them better within the tree. I just briefly want to talk about the genus names here very briefly. So for one of them, we called it Galosia, which is named after a Phoenician trading ship. And the other one called Lembos, we called after a fast Illyrian galley. Let me just do this so I can actually show you the video. Just for the uninitiated, which I assume is almost everyone except Alistair, Ploiecids come from a Greek term ploion, which means small boat. So our kind of tradition is to additional genus names will have a naval theme attached to them. So that's the reasoning behind this. So Galosia, you can see, actually has is pretty big, and if you look very, very closely, and I'm not sure if you can see this over the interwebs, has actually pellicle strips visible, whereas Lembosia is actually it's a bit smaller. It's a faster glider and has a really long posterior flagellum. It's also very granulated, so two very different cells. All right, so thank you, Alistair. These 5 additional single cell transcriptomes we were able to put into our multi-gene tree that I showed you earlier. So we basically have an updated version of our multi-gene analysis of euglenoids, and the new taxa in red here. And I'm going to talk about that briefly. But first of all, we do again find euglenoids as a monophyletic clade up here, and we sister to them. We have kinetoplastids, diplonemoids, and symbiontids, and then at the bottom we have the outgroup. I'm just going to cut that off because we don't care about them right now. And so again, we can see, if we zoom in on euglenoids, we can see that we recover the Spiracutidae clade, which again includes phototrophic euglenoids and a whole bunch of other phagotrophic euglenoids. At the bottom, again, the most basal branch of euglenoids that we know to date are petallomonads in orange. And kind of in between, again, those two in the mid backbone of the tree, so to speak, we do recover ploiecids, or a loose assemblage of ploiecids. Okay, so the first cell I want to talk about briefly mention is Decastava. This is one of the cells where we had SSU data before, but now we have multi-gene data. And it turns out this Decastava cell actually falls within Alistosa, which is a previously described group of ploiecids. This is not too surprising. We kind of knew this would happen from the SSU phylogenies alone, but it's nice to have this multi-gene data now. The second one I would like to talk about, 2 cells, are Hemiolia, and it turns out in all of our phylogenies they go sister to a taxon called Liburna, and you have a video of Liburna here and a picture of a different Hemiolia. They're always go together with very high support. And it turns out that actually kind of makes sense from a morphological standpoint as well. Turns out Hemiolia and Liburna look very similar, as you might be able to tell, but their movement pattern is the thing that really sticks out, to be honest. So Liburna, as you can see in the video, it glides very fast, usually in an arc, but then it stops and just hangs out for a while, and then it continues. This doesn't sound particularly weird, but in the grand scheme of euglenoid movements, that's actually kind of odd. So this appears to be the only group where that is happening. And because of the morphological similarities and the phylogenetic affinities of those 2 taxa, we decided to establish a new clade called Caravia, which is named after a Portuguese sailing ship, a caravel. Okay, so second last cell I would like to talk about is Lembosia, which you can see up here. And in all of our phylogenies, Lembosia goes sister to a taxon called Alcasia, and you've seen this video earlier. You can see it again here. We recover this always with very high support, but if you look at the morphologies, they don't really look that similar, at least judging by the light microscopy data that we have available. You can see Alcasia is a pretty big cell. They can get up to like honestly 60, 70, 80 micrometers, which is big for a phagotrophic euglenid. And you can see the pellicle strips clearly, whereas Lambosia, again, much smaller, so maybe 20, 25 micrometers, and is very granulated. So not sure what's going on there, but it is a relationship we recover routinely in all of our phylogenies. Last but not least, we have Galosia, which you can see an image of again here. And this is the only taxon where, you know, it kind of goes nowhere in particular. So it's definitely a polychaete, but in different analyses, so just using slightly different models or slightly different taxon sampling, this taxon actually jumps around within different polychaete subgroups. So its exact position is not entirely clear from the data that we currently have available. So that's just something to keep in mind here. And I will say the amount of data available for this multi-gene analysis is likely not a limitation in this analysis. If you look at the amount of sites available for Galosia, it's actually very good. So all of this, again, leads me to believe that there is likely, if you look at the mid-backbone of the tree, that there is likely additional, diversity out there that if we were to sample and do single-cell transcriptomics on them and do multi-gene analysis out of those transcriptomes, we might actually be able to resolve this particular part of the Euglenoid tree. And with all of that, I would like to thank all of you. And particular shout out to our sequencing center. They do amazing work and actually really teasing out the last little tiny sequencing bits out of the single cell transcriptomes. Thank you.
Thank you very much, Gordon. Our last presentation is going to be a video presentation. Is that right, Scott?
That's correct. We're going to load it up now.
So the presentation is by Rio Harada on DNA polymerase in Euglenoid plastids. So hopefully this will now magically appear and we can...
Yeah, we're just going to load it up here. Thanks. 1 second.
We can't hear any sound, or I can't hear any sound.
Just 1 moment.
Hi everyone, my name is Ryo Harada. I am a PhD student in University of Tsukuba, Japan. And first of all, I'm sorry for not being able to attend the meeting and presenting via video. Today, I will talk about the plastid localized DNA polymerase of Euglenoids and its origin. So let's start with an introduction. Organella localized DNA polymerases. I have been studying this protein, and today's presentation is based on a part of that work. Organella localized DNA polymerases are encoded in the nucleus and are required for the replication and maintenance of mitochondria and plastid genome. And organella localized DNA polymerases have sequence similarity to the bacterial DNA polymerase 1, POL1. And there are several types of organella localized DNA polymerases which have independent origins. The most popular type is POP, which is localized in either or both of mitochondria and plastid of broad eukaryotes. And the DNA polymerase gamma, called gamma, is mitochondrial localized DNA polymerase of opistoconta. And PREX is a plastid localized DNA polymerase of apicomplexar and clonpoderids. Finally, POL1A and POL1B3+ are the mitochondrial localized DNA polymerases of Euglenozoa. These types are the organella localized DNA polymerases found so far, but in some eukaryotic lineages, there are organelles whose organella localized DNA polymerases are unknown. So this means that there are still unknown organella localized DNA polymerases. Therefore, I'm researching the diversity and evolution of this protein. So today's talk is about the plastid localized DNA polymerase in Euglenoids. But before that, I will review the secondary plastids in Euglenoids. This figure shows the maximum likelihood tree of 70 plastid coding genes. This figure shows the plastid of Euglena is closely related to the plastid of Pyramimonas, and suggesting that the plastid of Euglena is derived from secondary endosymbiosis with Pyramimonas. And this is shown on the eukaryotic tree. And Euglena belonging to Discover acquired plastid by secondary endosymbiosis with Pyramimonas, which is belonging to the green algae. So here, the plastid localized DNA polymorase of the green algae is shown. And Pyramimonas also has a pop. However, the plastid localized pop was not detected in Euglena and Euterptera in my previous research. Therefore, Euglena and Euterptera may have a novel type of plastid localized DNA polymerase. So we searched and classified the DNA polymerases in Euglenids and green algae. Here are the results. We found two types of novel DNA polymerases from Euglenoids and Pyramimonadeus species, respectively. From 4 species of Euglenoids with plastids, namely Euglena and Euterptera, we found Eugpol-A. And we also found the Pyramipol-A from 4 species of green algae, including Pyramimonas. These two types were closely related in phylogenetic analysis for classification, but did not form the sister relationship. The subcellular localization of Pyramipol-A is unknown, but Eugpol-A of Euglenoglacylus was detected in the proteome of plastid. In addition, Eugpol-A was not detected in Euglenids without plastid, such as Raphaza, Peranema, and Rhabdomonas. So now we can conclude that the plastid localized DNA polymerase of Euglenoids is Eugpol-A. Next, we performed a detailed phylogenetic analysis to confirm the phylogenetic relationship between Eugpol-A and Pyramipol-A. In this analysis, Eugpol-A, Pyramipol-A, and their closely related bacterial DNA polymerase 1 were used for maximum likelihood and Bayesian inferences. The right figure shows the maximum likelihood tree. The maximum likelihood tree and Bayesian tree had almost the same topology. And blue nodes show the eukaryotes, and red shows the gamma proteobacteria, and yellow shows the beta proteobacteria. As a result, the Eugpol-A and Pyramipol-A, most of these DNA polymerases form monophyletic clays, and these clays form a sister relationship with bootstrap value of 64 and Bayesian posterior probability of 0.99. And these eukaryotic clays were closely related to gamma proteobacterial clade. This result indicates that the Euglenoid A and Pyramid A share the same origin, and which is closely related to the gamma proteobacterial DNA polymerase 1. So, integrating the results so far, now we can assume an evolutionary scenario for Euglenoid A, a novel plastic localized DNA polymerase of the Euglenoids. The previous phylogenetic analysis shows that the Pyramid A was closely related to the DNA polymerase 1 of gamma proteobacteria, and indicating that the Pyramid A is derived from the gamma proteobacterial poly 1 via lateral gene transfer (LGT). And second, the Pyramid A and Euglenoid A are sister relationship and share a common origin. And considering the secondary endosymbiosis of pyramid monas and euglenoids, Euglenoid A may originate from Pyramid A via endosymbiotic gene transfer (EGT). Of course, the possibility of independent LGT from bacteria to these eukaryotic lineages, but our scenario is the most parsimonious and makes sense. This is the final slide. First, we identified the novel DNA polymerases called Euglenoid A and Pyramid A from species of Euglenoids and Pyramid monodales. And second, Euglenoid A was detected in the plastid proteome of Euglenoids. So Euglenoid A is a novel plastid localized DNA polymerase of Euglena and Eutreptera. And third, based on the results of detailed phylogenetic analysis, we proposed an evolutionary scenario for Euglenoid A through LGT and EGT. And below are the acknowledgements. This work is supported by these grants of JSPS and supercomputers in DDBJ. Thank you for your listening, and I hope to see you at the next meeting.
Thank you very much, Disembodied Rio. That brings the session of presentations to an end. We have about 15 minutes for any questions that you have for Marek or for Gordon. If you have any pressing questions for Rio, I'll pass them on when I see him at dinner this evening, because he happens to be in Halifax right now. Yes, please just type them into the chat. We have one question to Marek from Vladya, with a partial answer, I think, from Zoltan. But if Marek wants to comment any further on this, please do so. You mentioned multiple proteins translated as polyprotein and processed in the plastid after import, like RBCS. Is there a visible common motif between the units for all these cases that could be recognized by the same protease?
Yeah, so just briefly, so the previous literature did establish some similarities between the motifs or the structure of the short peptides separating the units in RBCS polyprotein and the LHC polyproteins. So yeah, this suggests that there might be the same protease that is responsible for cutting or processing the polyproteins. Whether the same holds for those extra proteins that we have identified, honestly, I don't remember. I think we did look into it. Maybe Zoltan remembers better than I do. I think the length variation in these putative linkers in those translational fusions combining different proteins, I think they tended to be of different length or varying length between the different fusions. So maybe the mechanism for the processing, which in fact has to be demonstrated, we just assume that they are split. after transporting to the plastid. So probably or potentially it might the processing might work differently from these polyproteins, but yeah, we are just guessing.
Can you remind us if they're the same in, I can't remember from the slide, but are the polyproteins seemingly identical in the two species of Euglena, or do you see?
Yeah. Oh yeah, I did not comment on it. Yeah, so yeah, we frequently find the same fusions in at least in the different Euglena species. On a slide, I don't remember whether we have checked their potential like broader conservation. At the time we did the analyses, there were very little data from other euglenophytes, maybe Eutrepsiella, but the assembly is not of, or the assemblies are not of that good quality. So I don't know whether these are conserved, but at least between the two species, they tend to be conserved.
Thanks, Marek. While we have you here, I just was wondering if you had any speculation on how the second of the three membranes around the plastid might be crossed if there's no TOC there.
Yeah, so I think Vladia is certainly more competent to answer this question, but we did in that paper with Anna Novak-Vanslova as the first author, we did provide some a potential answer. So basically, that euglenophytes specifically and code for proteins that are homologous to so-called derlins, are basically proteins that are components of the ERAT complex at the endoplasmic reticulum, and in fact also the SELMA complex that is involved in traversing the second outermost membrane in red algal-derived secondary plastids. So that these might be good candidates for for proteins that are implicated in this transport step. But whether this is the case, of course, has to be demonstrated experimentally. So maybe Vladia I guess Vladia has a better answer than I do.
We now wait to see if Vladia will.
Oh, okay. So should I? Well, I have nothing. Yeah, I have nothing more to add. Yeah, this is still our working hypothesis. We are going after it. We haven't localized it, but yeah, we will see.
Thanks. We have a question from Gordon, I think from Vladia as well. He asks, is the type of active gliding flagellum, either anterior or posterior, a phylogenetically informative character in phagotrophic euglenids?
Yeah, that's a good question. I think the answer is yes, with some caveats. But in Pluischids, for example, like all of them glide on their posterior flagellum, and the anterior flagellum is used like solely for feeding. Kind of hard to tell, but it usually like flips around in the front, and when it encounters a bacterium, for example, it kind of like curls around and then pulls the cell closer. For example, in petallomonads, you do have, petallomonas and notocoelinus. Notocoelinus supposedly has two flagella. There's some taxonomic, issues with that. But both of them appear to glide on their anterior flagellum. And if you go within Spirocheta, for example, there's a lot of taxa that just have a single flagellum. And those obviously glide on the anterior flagellum because it's always directed to the front. Yeah, it's kind of a tricky question because I assume the more taxa we sample, the more complicated the picture will get. But there's also indication that there are a couple of taxa, including Heteronema, certain kinds of Heteronema that might glide on kind of both flagella, but it's a bit tricky and a bit tricky to test as well. So yes, you can tell, but you can also kind of tell just based on morphology where things fall at that point anyway. I hope that kind of answers your question. That was a bit roundabout way of answering.
We have actually another quick question for Gordon from me, if that's okay. Would you like to briefly comment on? I know you were looking at the phylogenetic at the single cell transcriptomes for phylogenetic purposes, but could you briefly comment on how good those single cell transcriptomes are from those 5 taxa, and whether, to what extent that data is useful for other purposes like gene discovery? So I guess how complete and how uncontaminated are those new transcriptomes?
Yeah. So for the multi-gene stuff, they're pretty decent, actually quite good. It's quite a bit of a range. Out of the 5, there's one that's kind of borderline okay, and the others are pretty decent, one of which Galosi, I think, is actually really quite good. In terms of how complete they are, I should look at the BUSCO scores again, but they weren't crazy high. I would say like 50% complete max, which for a single cell transcriptome is actually not terrible. It's actually pretty good. And there's some discussion whether BUSCO scores are a good proxy for transcriptome completeness in these, how good that actually is in these groups anyway. But I personally haven't looked into discovering any other genes in the transcriptomes, but it's likely something I will probably get around doing at some point in the future. So I assume they're pretty good, but that being said, I haven't really looked at them that closely in that regard.
What about contamination?
Oh, contamination. Yeah. We did run blob tools for a couple of them, and they do look pretty clean. There's obviously always a bit of like, prey contaminants in there. And you do get the occasional, bacteria, but considering it's a eukaryotic transcriptome that was made with like an mRNA selection step, the amount of bacteria is pretty limited. So, yeah.
Thank you. We have a question from Dr. Nakazawa for Marek. You mentioned about some fusion proteins in Euglena longa and Euglena gracilis. Are there any ideas how this unique fusion occurs in previous evolutionary events? So I guess the mechanism for how those fusions arise.
Yeah. So I'm afraid I don't have any like specific answer. I would say that these fusions are not any different from any other similar kinds of reshuffling of genetic modules, like individual protein domains or acquisition of proteins of organoid targeting pre-sequences. So these are probably things. So simply pieces of the genetic material reshuffle all the time, and time to time, they happen to make a meaningful match, and these are kept, and that's it. So I don't think that there is any specific, or at least I see no indication that there would be any specific mechanism for making this. Of course, the question is why we see this in euglenophytes while not necessarily in other groups. The question is whether this has been explored to any sufficient detail in other groups. Maybe it has just not been noticed. Maybe it was just has been missed in other groups, and it's more common than we think. So yeah, that's what I can say.
I have a terrible hypothesis for you.
Yeah, go ahead.
My terrible hypothesis is that the plastid import is hideously inefficient when you don't have TOC, and therefore there's a greater pressure to streamline the plastid import system in other ways. Yeah, maybe it has something to do with the specificities or specifics of the plastids. Maybe it has something to do with the specific aspects of the genome organization in euglenophytes, with all these unusual introns and so on. Yeah, so who knows? It's extremely difficult to guess. We can only speculate, I'm afraid. Yes. Also, do I remember correctly that the different polyproteins, they are functionally related, are they? Or are they? So in these polyproteins, like rubisco small subunit or LHCs,
these are basically just repeating the same thing multiple times, and the repeats are more or less identical even at the nucleotide level. There are there might be some variation in E longa, for example. The Rubisco small subunit polyprotein is more, so that includes units that appear to be disrupted. So only some of them are probably functional, but these are the same proteins. While we also see these fusions of different proteins that have nothing in common in functional terms. So both cases are possible.
Not even in the same pathway.
Yeah. So you saw there was, I think, a superoxide dismutase fused to a metabolite carrier, so a transmembrane protein. So they are most likely they are split and function independently. Yeah.
I'm sorry, Dr. Nakazawa, did you have a comment?
Thank you. And I wonder, you're going to have also a very unique fusion enzyme also in the mitochondria. So I wonder about the question. Yeah. Thank you very much.
That's interesting. Okay, thank you.
I think we're at the end of our time now. So please join me in thanking all three speakers very much, and thank you for a fascinating discussion there. Really appreciate it.
Thanks, guys. Thanks, Alistair.
Thank you very much, and yeah, see you. Bye-bye.
Thank you, Alistair. And I'd like to introduce, it's now time for our new award this year. So that's the Best Paper Award, and that's sponsored by Kemin. So I'm going to invite Jeff Horst from Kemin to make that announcement and present the selection. Over to you, Jeff.
Great. Thank you, Scott. Can everyone see my screen?
Yes.
Okay. So yeah, thank you very much, Scott, and the organizers for the conference. Kemin is very happy to help support the conference. And so yeah, we have two different awards this year. One is going to be for Best Paper, and then tomorrow we're going to have a second award for Best Photo. And so there was 7 submissions for this year's Best Paper. And actually, I read all of them myself as well, and lots of great research going on, really complementing the research that we're hearing about during the conference this year. But there can only be one winner each year. And so we're very happy to award this year's Best Paper to Mohammed Al-Dami and his colleagues on a paper about euglenoids. And so congratulations to that team, and we'll reach out to you on the cash award. So thanks again. And tomorrow sometime, I'm not sure the exact timing, will be the photo award. And then for the rest of the folks, we'll also be having a technology showcase at the very end of the day tomorrow. So we hope that you stick around and listen into all the stuff that Kemin's up to, dealing with Euglena and our immune products. So that's all. I'll let everybody move on now.
Thank you so much, Jeff, and congratulations to the award winners. New award this year, and we look forward to tomorrow's presentation of the best photo as well. So that brings us to the end of our second session and the end of today's conference. So I'd like to thank again the chairs and moderators and all the presenters, and we look forward to seeing everybody again tomorrow for our last day on translation and commercialization. We look forward to seeing you then. Have a great evening, morning, night, whatever time it is. We'll see you tomorrow, everyone. Thanks again.